This video will provide some examples of some possible final exam questions. And I'll try to explain these as well as share some PowerPoint notes and PowerPoint slides and some maybe pictures or graphs I've copied from our book or copied from other sources. So here are just a couple possible final exam questions. One exam question would be to make sure you understand the flow chart or the step-by-step process of muscle excitation contraction. And I would probably type this out or make some kind of flow chart and make sure you understand the correct steps or the correct pathway of this flow chart. And we would start with motor nerve depolarization. So that's at the site of the neuromuscular junction, where the nerve sends its signal by acetylcholine to the muscle cell membrane, and that's where the muscle cell becomes depolarized. And then we'd end with muscle contraction. So how do the actin and myosin proteins get together? So this would be the beginning of the pathway, at least in terms of our discussion. Or here's the neuromuscular junction. And here are these vesicles, synaptic vesicles that contain acetylcholine. And when acetylcholine goes into this synaptic cleft and binds to receptors on the muscle cell membrane, that initiates the wave of depolarization. That initiates the excitation at the muscle cell membrane. And I copied this picture from another textbook. And this whole picture is a muscle cell, and here's the muscle cell membrane, the muscle plasma membrane, or the sarcolemma. And this dashed line would be the muscle action potential that came from the nerve, but then was propagated along this muscle cell. And you should understand this picture and this process where this action potential goes along the membrane, but then dives down deep into the muscle cell by this transverse tubule, goes past the lateral sacs of the sarcoplasmic reticulum, and then somehow, not completely understood, but opens up channels that allow calcium to rush out of the sarcoplasmic reticulum. And as we've described before, calcium will bind to troponin, causes sort of a change in shape of troponin, and lifts tropomyosin out of the groove of actin, exposing a binding site on actin for myosin. And here's another picture from our textbook of this sliding filament model of muscle contraction. And here are the proteins of the thin filament, troponin, tropomyosin, and actin, and the myosin or thick filament at the bottom. This is a relaxed state, and obviously then this is a contracted state where the myosin head is allowed to bind to a binding site on actin. So you should be aware of this step-by-step process. Another question would be to describe, and what we've mentioned are three main muscle fiber types. So be able to describe those individual fiber types and the names and abbreviations for those fiber types. So the fast fibers consist of type 2B. They're fast twitch and fast glycolytic, so they do a lot of anaerobic or fast glycolysis. Intermediate fibers are also fast twitch. They're classified now as type 2X, and they do both oxidative metabolism and glycolytic metabolism, so they're intermediate fibers. And on the other end of this sort of spectrum are type 1 fibers that are slow twitch and do predominantly oxidative energy system metabolism. And be able to describe these fibers as well, like mitochondrial content or the velocity of shortening, the efficiency. And we had made a table earlier sort of describing these different fiber types. Another possible final exam question that's also related to muscle contraction, and I believe I've mentioned this before, is to list sort of the functions or jobs of this myosin head. And a lot is going on at this myosin head. So it has ATPase activity, so where the myosin head is able to break apart ATP into ADP and phosphate. And that allows the actin and myosin to slide over top of each other. If it has ATPase activity, it's going to bind ATP. So someplace on the myosin head is an ATP binding site. And it also acts, obviously, to bind to actin. So someplace on the myosin head is also an actin binding site, and that's how actin and myosin to get together and a strong cross-bridge formation occurs. So list 3 functions of the myosin head: ATPase activity, ATP binding site, and an actin binding site. Another protein involved in the sarcomere is the largest protein in the human body called titin. And here's a picture from our book that sort of shows titin. So here are 3 different sarcomeres. The top panel, the sarcomeres are the distance is greater. So the sarcomere length is 4 micrometers. And a sarcomere is from this Z line over to this Z line. And then there's one in the middle as well. So this would be a sarcomere. And then here's a contracted state in the middle and the most contracted state at the bottom, where the sarcomere length now is only 2.7 micrometers. Titin is shown just by this line, and titin sort of anchors the sarcomeres together. So it's thought of as sort of a structural component to keep sarcomeres together. And so here it is right here, and it can span the length of this sarcomere. It has also been shown that this titin protein has some elastic function. So part of the structure is sort of spring-like. And so now there's some suggestion that maybe part of the elastic or elasticity of muscle and of muscle contraction may be due to this titin molecule or titin protein. And there are slightly different forms of this titin protein as well, and that may change sort of the elasticity of muscle fibers and of whole muscle groups. So the titin protein. Talking about muscle contraction, and you've heard of this term before, so muscle hypertrophy, and hyper is big and trophy is size. So muscle hypertrophy is an increase in muscle size. And obviously, if there's a greater muscle size inside that muscle cell, then are more sarcomeres and more actin and myosin protein, so there's going to be greater strength. With resistance kind of training, we're not producing more muscle cells. So there's not really good evidence that we're actually growing new muscle cells. The ones that we have are increasing in their size. So it's going to be a greater muscle hypertrophy. Another good final exam question, maybe it's more of a short answer or essay kind of question. And the question is, how does the size principle help explain the lactate threshold? And this is a nice question because it brings in things that we've been talking about for the entire class. So it's talking about the lactate threshold and the rate of glycolysis. So that's glycolysis. We talked about the beginning of the semester. Lactate threshold we talked about in the middle of the semester. And toward the end of the semester, we talked about muscle contraction. And so that involves the size principle and a motor unit. So this next picture is from our book, and it shows it's a graphic representation of the lactate threshold. Now, we've talked about this before, or if we looked at on the y-axis blood lactate concentration, and on the x-axis it's some measurement of exercise intensity. Here it says a percent of VO2 max. And there's 2 lines on here. The blue line represents a more untrained person, and the red line represents a more aerobically trained person. And both lines show a lactate threshold or a lactate curve, where the blood lactate is relatively constant in easy exercise. But then at some exercise intensity, the graph goes way up. It starts to go way up. And so the question is then, what are the factors that are related to this lactate threshold? And we've gone over these before, and here's a list of them in this graph. So it could be a low amount of oxygen. It is a reliance on glycolysis, and I've mentioned that before, and I'll show my pictures again in the next slide. And what we'll talk about now, or in a couple of minutes, it is activation of fast twitch muscle fibers, and that does involve the size principle. And also it could be a reduced rate of lactate removal. So those are factors causing or mechanisms for this lactate threshold. Along with mechanisms of this lactate threshold would be to remember the different forms of the lactate dehydrogenase enzyme. And the two main forms that we've been talking about, one is called the LDH muscle form of that enzyme. And so that's the enzyme that would convert pyruvate or pyruvic acid to lactic acid. So this would favor the formation of lactic acid. So it's the LDH muscle form, LDHM form. The same name of the enzyme LDH, or lactate dehydrogenase, but a slightly different form. It was originally found in heart tissue, and it's also now found in type 1 oxidative muscle fibers. And it would take lactate and change it back into pyruvate. And so one mechanism for this lactate threshold would be, which form of LDH enzyme is present. And I talked about that the rate of glycolysis, and I tried to use an example of a garden hose, and that pyruvate is sort of as a crossroads. It could go into the mitochondria or it could go out to lactate. And so the rate of glycolysis, how quickly we're breaking down glucose for energy, is a mechanism that's associated with this lactate threshold. You're working out at such a high exercise intensity, you need more energy, so the rate of glycolysis is faster. This next picture I copied from another textbook, but it simply lists sort of as boxes these different factors that can be explaining this lactate threshold. And so now the one I want to focus on, and that will help answer that question about the size principle, is in this box, that is the recruitment of fast twitch fibers. And you now can describe what are fast twitch fibers, and you can group sort of the 2B and 2X together. Obviously they're they're fast contracting, and they do a lot of glycolysis, right? So they wouldn't do much oxidative metabolism, they do more anaerobic glycolysis metabolism. And so they have we predict they have more of that LDHM form of the enzyme. So these are, if we would describe, those are sort of those fast twitch muscle fibers. So let's go into that question. So how does the size principle help explain this lactate threshold? Well, one thing to understand is what is a motor unit. So a motor unit is the nerve. So it's a motor neuron and all of the muscle fibers that it innervates. In my next slide, I just copied up sort of a picture of this. So it's one nerve going to many different muscle fibers. So the size principle is motor neurons that are relatively large. So motor neurons with relatively large axons, so large nerves, they become recruited as muscle force increases. So if you're doing an incremental exercise test, like what we showed with that lactate threshold graph, and now the work is getting harder and harder, you need to recruit, or there's more demand for force generation, so you're going to recruit more muscle fibers. So according to the size principle, you're using these larger axons as muscle force, recruited as muscle force increases. What helps explain the lactate threshold is that these larger size motor units innervate mainly type 2 fast twitch muscle fibers. And you can describe those. They're glycolytic, they're fast contracting, they have low amounts of mitochondria, so they're going to be lactate producing fibers. So this next picture I found online that is it's a picture of a motor unit. So what's stained in dark is the nerve, the motor neuron, and then these lighter stained tubes are the actual muscle fibers. So here's one nerve, and you can sort of see where it's connecting to all of these different muscle fibers. So this would be in a picture example of a motor unit. And so as I've described at the beginning of exercise, slow motor units that are smaller in size, that innervate, they do innervate predominantly type 1 fibers. So these are oxidative fibers. We're not producing much lactate. There's not that great demand for energy and demand for force production. But as the exercise intensity increases, now we need to recruit additional motor units that innervate predominantly type 2 fibers. The exercise intensity is hard. We need more force generation, so we're going to recruit these type 2 fibers. And you can describe these type 2 fibers. They're glycolytic, they're lower mitochondria, so an increase in the rate of lactic acid production. So this next picture is from our book, and so they just show the number of active motor units on the y-axis and just a measurement of exercise intensity, so light, moderate, and heavy on the x-axis. And so to start off with, in the blue line, it's predominantly type 1 fibers, and the exercise intensity is light, so we don't need to be recruiting that many muscle fibers. Yet when the exercise intensity gets heavy, we're pushing so hard on the pedals of the bicycle. We're swimming hard. We're running hard. We need greater force generation, so we're going to recruit additional muscle fibers. These additional muscle fibers and the motor units that they're coming from are larger size motor units that innervate, that connect to these type 2 and type 2B or type 2A, 2X fibers. And these are fast contracting type 2 fibers that are lactate producing fibers. So this is a nice question because it explains several things that we've talked about in class. The rate of glycolysis, a lactate threshold graph, and now the size principle, a motor unit, and then describing what are type 2 muscle fibers. So I hope these couple questions are good practice for some examples of final exam questions.